planet we live on, a rotating mass of rock and water orbiting the Sun at around 106,000 kilometers per hour, is so much more than just a surface that we live on. It is a complex system teeming with life, and in fact, it makes important physical contributions to the life forms that exist on it. The water that falls and flows over the Earth's surface, and even the rocks in different geological zones, contain elements such as oxygen and strontium respectively, which have characteristic regional signatures in the form of isotope ratios. These elements find their way into the water we drink and the food that we eat. In fact, they are incorporated into our body tissues, bones, teeth, even hair, and fingernails as they grow. This means that we retain the signals of the place where we were while these tissues were forming throughout our lives. As the isotope ratios of these elements vary from place to place, depending on rainfall for oxygen and geological substrate for strontium, it is even possible to retrace people's lives and movements long after they are dead. So, how does this work? Archaeological skeletons can provide a lot of information about the lives of past human populations. When studied by anthropologists, health conditions and diseases can be determined by identifying specific pathologies on the bone. It's also possible to discover behavioral patterns, such as everyday occupational movements, that cause the strengthening or weakening of bone in certain areas, resulting from repetitive muscle use. Although the surface of the skeletons can already tell us a lot about people's lives, if we dive even deeper into the elements that make up those bones, we can dive deeper into the past. Taking strontium as an example, the journey of the atom starts in the magma that becomes bedrock, eventually breaking down into the soil in which we grow our food. The atom travels through the plants that grow and the animals that eat them, all the way to the food of a prehistoric person and then into their bones and teeth. Calcium and phosphate are the main elements that make up the apatite portion of bones and teeth. However, sometimes elements that are chemically similar to calcium, such as strontium, barium, and even lead, can sometimes substitute for calcium atoms in the body, which is how it becomes incorporated into the growing skeleton. This strontium is finally released from the skeleton in a laboratory and can be quantified using mass spectrometry. The strontium isotope ratio is a value that reflects the average of all strontium that contributed to a sample, from all the strontium atoms and their various sources and journeys that came to be in the bones and teeth of a person. Teeth form at different stages of childhood. First molars during 0 to 2 years, when a child is usually breastfeeding, second molars between 3 to 6 years old, and third molars between 9 to 12 years old. This means that careful tooth selection is important, as the information obtained will reflect different phases of life, and in doing so will answer different research questions. Once teeth have been selected, they need to be cleaned to remove external soil particles from the burial environment. We are interested in the isotope ratios of strontium and oxygen stored in the tooth enamel itself, which is dense, hard, and highly resistant to diagenetic chemical changes. For this reason, it gives the best chance of reflecting information from the life of the individual. Loose soil is mechanically removed with a drill so that it does not contaminate the enamel sample and influence the results. After the external part of the tooth has been cleaned, the drill is used to remove enamel powder, which is then collected and weighed before being chemically treated. The chemical treatment of tooth enamel is necessary because of post-burial contamination. While this is a bigger problem in bones than in teeth, due to their greater porosity, groundwater strontium and oxygen can potentially replace some of the in vivo elements in the pore spaces of enamel. The diagenetic carbonates can be removed relatively easily by cleaning with a weak acid solution, as they dissolve more easily than the original dietary strontium, which is more strongly bound to the calcium site in the apatite. After treatment, the isotope ratios of the elements of interest can be obtained with mass spectrometry. The strontium isotope values can tell us about the geological region where a prehistoric person lived and sourced their food, while oxygen isotope values can tell us about the drinking water. When these values are compared to predicted local values for an archaeological site, it becomes possible to identify local and migrant individuals in an ancient population. Predicted local oxygen isotope values can be calculated from precipitation values in a particular region, whereas local strontium values are best estimated from averaging the values of animals found within the same archaeological context, because they were likely to eat food grown in local soils. The ability to identify migrants and non-migrants within prehistoric populations from their skeletal remains is a very valuable tool, because such detailed information about the lives of specific individuals can rarely be found in historical sources. Modern advances in earth sciences allow us to access the kind of detailed information that was simply unimaginable in the discipline of archaeology just a few decades ago.